Thank you. It's a pleasure to be here. So I'm a lapsed computational neuroscientist in the philosophy department of Oxford University. And one reason I got into philosophy is that I became interested in issues about futuristic questions about how can we enhance brains, how can we think better, how will new technologies affect the world. So I ended up at the Future Humanity Institute. So we are kind of a crazy part of a already rather strange philosophy faculty. So we're trying to look at big picture, long range questions that would change the situation of humans in a big way. So one area of our research is global catastrophic risk. How could things go terribly wrong? Another area is rationality and epistemology. How can we actually think sensibly about future things where we have very little information? But we're also interested in emerging technologies that if they develop would really change the rules of the game. And as a nice intersection between both my background and this, we have this field of whole brain emulation, which I'm hopefully going to give you a little talk about our estimates and some of the consideration about the technology that we have been thinking about. So the idea of doing a whole brain emulation is really kind of bringing the classic philosophical and science fiction plot device of making a one-to-one copy of a brain into reality. We have a beautiful existence proof that we can have intelligent systems in this universe in the form of our own brains. And obviously we want to learn interesting things from it. The interesting question to a philosopher might be, of course, could we actually get minds out of our skulls? Could we actually, by copying the neural structure of our brain, actually create a piece of software that was doing real thinking? Whatever real thinking actually is. And here you can imagine a gaggle of philosophers going on for quite a long while about that question. But you can also view it as a pretty interesting technical question. So one reason we started thinking about this is it's a good exercise in forecasting. Artificial general intelligence is another really powerful technology that would really change everything. But we have no idea about how to estimate timescales. We can actually, as I hope to show, make some rough estimations about some of the timescales for brain emulation. So this makes it a fun exercise in forecasting. And of course, that means, of course, that in 50 years' time, we're going to be laughing quite happily about it too, all the things we got wrong. But it's a well-defined problem, and understanding that might be very useful. An interesting thing is also that making a one-to-one copy of a technical system means that you don't necessarily need to understand what's actually going on in there, why it was designed that way. And that seems to be very different from the normal procedure in science, where we try to understand a complex system, usually by making very simplified models of it, and then we get an understanding. Here it's more about low-level reverse engineering. And finally, another reason is that if this happens, if we can actually make a good brain emulation, it would be a very big thing. Obviously, from a kind of philosophical standpoint, if you could finally upload a philosopher and ask him, are you really conscious? Do you feel yourself to be you? That would help perhaps answer some interesting philosophical questions. Especially, of course, if you took the skeptic, uploaded him and said, oh yes, I actually think I'm conscious, I was wrong. That would be really useful. The big problem would be if you upload and says, no, actually, I'm not conscious. So that's going to keep the philosophers busy. And of course, from our neuroscientific perspective, it would also be interesting, because this is kind of a logical endpoint of neuroinformatics, getting all the relevant data existing in the nervous system into a computer-readable form. But if you could actually run human brain emulations, it would also have economical impacts which are downright frighteningly large. The economist Robin Hanson has written one scary little paper where he demonstrates the consequence of getting copyable human capital and essentially get an economic, if not a pure mathematical singularity, for all practical purposes, having an economy with a doubling time corresponding to Moore's law, well, that's still significant enough. And that means that we actually want to figure out how possible is this, and ideally do that a few decades ahead. Because suddenly getting an economic singularity when you don't expect it is probably going to be pretty bad for the geopolitical stability of the world. So we did a little workshop a while ago at Oxford where we invited experts in various fields together. We banned all philosophical discussions. So no discussion about what consciousness really is or what software actually can represent. Just the questions, what resolution of brain scannings do we need and what methods could work? And then we got started in digging into that. And Well, what about emulations? Besides that, my dear ZX81, my first computer, now nicely covers the title. The idea is, of course, old-fashioned in computers. We can actually emulate both hardware and software in another medium. So we want to replicate the causal structure of what's going on inside one system in another one. A simulation just appears similar to what is being simulated. That might actually be well enough. We don't really want a weather emulation that emulates all the molecules moving around in the atmosphere. That would be simply overkill. We just want a weather simulation that's good enough to predict whether it's going to rain tomorrow or not. So this obviously exists for computers, but computers have a lot of properties which are very nice for emulations. They generally tend to have well-defined abstraction levels, and they tend to be very discrete systems. An interesting question is whether the brain is actually behaving according to that. And that leaves us with some interesting scientific questions, which are again good for job security among neuroscientists, but also actually pretty important research questions for other purposes. As I'm hoping going to be able to demonstrate, this is going to synergize a bit with other neuroscience. So the basic idea is really building a brain without understanding it. So if somebody gave you the schematics of a microprocessor and you happen to have a factory that could make silicon chips, it doesn't matter that you have no clue what the different parts are doing. You could ideally, if you're skilled enough as a fab engineer, replicate that processor. And the idea of brain emulation is, well, this might be possible for real brains. Most research about the brain is, of course, attempting a high level of understanding. We want to know what memory is, how it does. We want to understand what intelligence is. But that's a high level, rather complex concept. It's much easier to know what chemicals are around, how many different neurons of a particular type are there, how are they connected to each other. So instead of moving towards a high level of understanding of function, this is about getting a large understanding of details. So this is a bit more of a brute force approach. It's a bit like the Human Genome Project, where instead of trying to figure out in each individual gene what we're doing, first we sequence the whole DNA and then we hope we can understand it. In this case, there is the hope that, well, if we have a kind of sequenced the whole brain, we would be able to run something very similar to it. So we would need to understand a bit of function. What does a neuron or some other component of the brain do? How can we actually simulate this efficiently? That, of course, leads to an interesting set of problems. First of all, we have a lot of things we don't know, and we know that we don't know it. I'm going to get back to the question of scale. Actually, what resolution would we need to simulate the brain on? And we also need to make certain assumptions. Some of these ones are kind of philosophical, like, well, we haven't got a metaphysical soul that's actually behaving separately from the brain. Although a really good brain emulation might actually be a very nice test of this theory. You could compare the brain emulation, presumably lacking soul, and see the differences in its behavior from a brain having a soul, and that might actually allow you to detect something. Most dualist philosophers would immediately point out that they can find a sneaky way around that anyway. Similarly, we of course have an interesting possibility that, well, maybe the brain is doing something non-Turing computable. Again, that's something we would probably want to know from a purely scientific perspective and perhaps even an engineering perspective. If there is a system around that is doing interesting non-Turing computation, we might want to copy that. Then we have, of course, issues about scale separation, which I think is interesting. Let me jump ahead here. I think scale separation is going to be kind of a key problem. And so if you want to have a really good scientific problem with a lot of applications in other fields to chew on, this is it. So we know from statistical mechanics that if you take a bunch of molecules in a gas and allow them to bounce around, their behavior averages out very, very nicely into macroscopic quantities like pressure, temperature, volume. On the other hand, if you have a turbulent fluid, the local scale behavior, all those little whirlpools produce in a turbulent flow, they don't average out. The low levels interact with the slightly larger levels, which interact with the even bigger levels, and vice versa. There is no nice separation. In a statistical mechanics, we can safely ignore the individual molecular dynamics because we have that scale separation. In turbulence, we can't do that, and that's also a big problem in simulating turbulence. The big question, of course, is the brain like turbulence or statistical mechanics or somewhere in between? Ideally, we would like to find the levels of scale separation. And if we could find methods of determining whether it's scale separated, that's obviously useful in a lot of other systems where we might want to engineer them. We want to figure out beforehand, is this going to be hopeless to simulate or not? So I think this is kind of a big scientific problem to chew on that exists in this neighborhood. But going back to assumptions here, we have a lot of other stuff which most of it should be pretty obvious. Similarly, we might also discuss the degrees of success, because we might still be pretty happy if we made a complete scan of everything that's in the head. And we understood that in a sense that we had a list of all nerve cells and their connections. That might be a very good treasure trove for science. That might be very, very useful for medicine. Similarly, that would be a parts list. We might also want to have just a very good scan for a lot of other purposes. We could imagine putting that together into a kind of functional database where you can run things, although it wouldn't actually produce very much. Then you could imagine a functional brain emulation. That is, you get something that produces a bit of plausible dynamics. You get brain waves, although you don't get a behavior. So it would be the parts are interacting at least in a plausible way. The chemical systems don't immediately go haywire. Then you could imagine something even more interesting, that you would get species typical behavior. So if you imagine that we do this with a mouse, you would get a piece of software that ran around and simulated labyrinth, hunting for simulated pieces of cheese in a good way. But it wouldn't correspond to the mouse you were originally scanning. So if you had a particular mouse and trained that for particular things, it might turn out that those things do not carry over. An even better level of success would, of course, be that it's an individual brain emulation. And it's at this point, of course, we start thinking that it might be interesting to humans who want to escape their bodies. And then we have a really tricky level, top levels, that it would actually have a mind, that it would be a continuation of personal identity, and all the other tricky social and philosophical stuff. But I think from a kind of engineering standpoint, we have to start down there. And then it's an interesting question how rapidly we would be moving up. I personally believe that if you manage to get a brain that actually does something species typical, it's going to correspond quite exactly to the individual brain we scanned. But right now, we don't have a really good scanned brain. We have, for example, a complete map of the nervous systems of a little nematode worm C. elegans. All 945, I think, neurons there are in this nematode worm. That's very nice, but it doesn't correspond to a single particular worm. It's just the general connectivity. And the really embarrassing part is we don't even know the sign of some of the synapses between the nerve cells, which I think is a real scandal, although it's very tricky to do this kind of research because those worms are darned small. This is, of course, why you want to run a test animal which has conveniently large neurons, which ideally are color-coded for our convenience. Well, what level of detail would we need to actually scan or process a brain in order to do something useful? Well, at our workshop, we got this very nice scheme that you can imagine actually several different levels that might be relevant, and we don't really have much of an agreement of which would be most relevant. So some people think that, oh, it's actually all about logic. We could perhaps do just the computational modules. That's a very, very high level understanding. That would require us to understand what memory and intelligence actually were and how they were in their processing. Then you can imagine a real low-level thing. Well, just run all the atoms. Do a big molecular simulation of every atom in the head. That doesn't require very much new information. We probably don't need any weird physics. We just need to know where all the atoms are located in somebody's head, and then a very, 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 very big computer to run it. And if we're really unlucky, of course, we need to take quantum stuff into account. And in that case, we'll need a very, 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 very large quantum computer. Hopefully, and this was the kind of rough consensus shown by raising hands, we're probably in the middle. Quite a lot of people think that, well, we probably need to model individual neurons. Because we know that you can train rats, for example, to respond to stimulation of individual neurons. If they get a reward whenever they manage to fire a particular neuron, or whether if you send a signal to a single neuron, just randomly selected somewhere in the brain, and you can train them to recognize that, oh, I got a signal, now I'm going to get a reward if I go to that part of the cage. So you can kind of have that kind of sensitivity in the system. Normally, of course, we don't care about our individual neurons, but we can kind of train ourselves to do that. So we probably might need a lot of chemistry. Obviously, the brain is not a pure, nice electric system. There is a lot of interesting chemistry going on. It might turn out that you can abstract this. So if some people like spiking neurons that get rid of a lot of the biological junk. It might turn out that you can actually simplify things further. Well, it's actually groups of neurons, not individual neurons that are doing the work. Or it could turn out that actually we need to care about individual molecular complexes, and then we're down there. As we go upwards in this diagram, we need more and more understanding about complex functional relationships. As we go downwards, we have simpler and simpler relationships. It's mostly physics and a bit of simple chemistry. But we need much more data and much more processing power. Perhaps if we had so good understanding of intelligence that we could be up there at level 1 or 2, we could probably run something equivalent to a human mind, probably on a laptop of today. I'm hand-waving wildly here, but Marvin Minsky made the same claim, and he's smarter than me, so I'm just leaving it at that. It's pretty clear that the molecular simulation of a brain, on the other hand, that's going to take something very different. Now, if we don't know which level we need to do this on, it seems to be an interesting research question, actually. And also, this is kind of a general method. You want to run simulation of different scales to compare each other. Because we have a problem with validation. In many engineering fields and science fields, we actually have a lot of work on figuring out, are we measuring what we want to measure? Does this actually work? In neuroscience, this haven't gotten started yet. And I think as we're going to see more and more kind of industrialized neuroscience, people are going to develop validation methods. And one nice thing is to have a bunch of very biological, realistic models, and then various levels of abstraction above them, and see that you can run them and get the same results. And then you compare that, of course, to your real biology. That way you can actually be a bit certain that you're not barking up the wrong neural tree. Well, with me so far. Looks like it. Well, scale separation was one of the problems. Another problem is, of course, that the brain is pretty messy. This is a typical electron microscope picture of how the brain really looks. You have probably seen all these beautiful animations where you see neurons suspended in space in some kind of big empty black space, and you have a neuron sending nice little sparkly signals to each other. That's, of course, the kind of science fiction picture that doesn't correspond to reality, because really everything is crammed together inside the tissue. So this, the stuff marked D down there, that's a dendrite, one of the receiving branches of a neuron. And then you have a bunch of S representing synapses, the points where neurons connect to each other. And as you can see, this is a slice through a very dense three-dimensional puzzle. Even worse, these synapses, they might come with different signs, in the sense that some of them are excitatory, they get the next neuron to fire it, and some of them inhibitory, they tell the other neuron to shut up. But they might also send other chemical signals, some of which change the behavior of the neurons. And this is very fun for neuroscientists, but it's also chemistry, and you can't really see the chemistry in this picture. So one interesting and outstanding question is, how much of the function can we actually infer from these kind of images? What kind of method of scanning do we need to actually be able to figure out what's going on in here? And it might look very, very messy here, but to some degree, of course, just finding the topology of this, especially when you got a stack of these images, that is just a large-scale image processing problem. And there are also some good signs that certain kind of synapses actually do have signs, visual signs, that they have slightly different functions. But as I'm going to show later on, there might be also techniques telling us a bit about the fun chemistry going on in there. Well, how much do we know about the brain? Well, that's another interesting question, because of this kind of project, you want to estimate how much there is. So here I plotted the time of discovery of neurotransmitters. And it started back in the 1930s when we found the acetylcholine, and then it started to ramp up. And now, for most kind of scientific progress, we would expect some kind of sigmoid curve. This one doesn't really look like a sigmoid curve. It's definitely not slowing down, or maybe it's slowing down, but I have reason to think, given that this paper was slightly old, that it might be underestimating what we discovered since 2000. But it doesn't seem likely that we would get orders and orders of magnitude more. We're still talking here about perhaps 150. There are good reasons to believe that, because there has to be genes and enzymes made, for producing these different kinds of stuff. And we know that the number of genes are finite. We can complicate things further, of course, because the body has found neat ways of making versions of enzymes. But again, it seems unlikely that most of our enzymes would be engaged in producing thousands and thousands of neurotransmitters. Similarly, we can look at the metabolism going on in a cell. Again, it's ridiculously complicated. But the number of different small molecules involved in our metabolism, I think, is of the order of about 5,000. Which is a very large network, and we're still not close to actually running a chemical simulation of them all. But there are pretty nice subsets we're already doing pretty good progress on. Similarly, the number of different kinds of neurons, again, it seems that depending on how you slice things, you might be getting anything between tens to a few thousands, or you might have very different weird clusters. Again, that we can do different scenarios about this kind of complexity, so we can actually estimate the computational demands. So it's an interesting challenge actually to do a bit of technology forecasting here, because we know there are unknown unknowns in there. And some of these ones are, we can say something. For example, the dynamical state of my brain in this moment. Suppose somebody magically scanned it but didn't take the activity level of the neurons into account. What would happen? And it seems very likely, given what we currently understand of neuroscience, that you would end up with a slightly confused version of me as you started the simulation, who've forgotten the last, at least a few minutes, probably a bit longer. And we have reason to believe this because some people are falling into really cold rivers and lakes in northern nations, and they've been revived after about 45 minutes. Now, neurons stop firing when body temperature goes below a certain level. So they literally had zero activity. They're flatlined in a very, very real sense. But that's not necessarily, but they could still reboot from this, so to say. And they still had the intact personality and everything else. So it seems that the dynamical state, that's not necessarily important. Maybe we need to scan the spinal cord, because we actually seem to do a bit of learning there. There might be other interesting problems here. Volume transmission, diffusion of chemicals. It turns out that that's computationally probably less expensive than actually running a neural network. Dealing with what glia cells, which previously people were just saying, oh, yes, they're just supporting the neurons who are the real stars. But it turns out actually that, well, the neurons are in the glia cells are actually, like any good servants and assistants, actually doing quite a lot of stuff. But again, they're doing it rather slowly, like most administrations. So it turns out that we can probably actually don't need to allocate that much more computation to it. There is a lot of fun stuff happening when synapses fire, because they adapt, they habituate if you fire too much. Sometimes we get stronger. It depends a bit on the chemistry. That's something we most definitely need to do. We probably need to do a body simulation, more about that later, to deal with the chemical environment. After all, our guts are actually containing a lot of neurons, sending a lot of chemical signals to our brain, and affecting our thinking quite a lot. Ghrelin, for example, a hormone which is involved in senses of hunger, is also a memory enhancer and actually changes our activity in frontal lobes. We tend to think of pictures of food as being larger, for example, when we are high on ghrelin. So if we want to make this realistic, we need to include that. But again, it's a rather slow dynamics, and it's not that terribly many things. Neurogenesis, the appearance of new neurons, that's slightly tricky. We have much less research on that. Again, a rather slow process. So if you left that one out, you would perhaps get a brain emulation that would have learning problems, but it would still be able to run a conversation. Epileptic effects, that's the weird electromagnetic effects that happen when you have two signals running on parallel axons. Then they actually induce magnetic fields that might synchronize them. And there are few people arguing that this is a real and important effect in the nervous system, and most of us who just say, no, it can't be that weird. It remains to see, actually. Quantum computation is, of course, the boogeyman to me. I don't think any neuroscientist really believes in it, but Stuart Hameroff does, and he's been arguing for that. Well, we're going to find out. It turns out that randomness and the ability to do analog computation doesn't seem to be that important. True analog computation is always swamped in the brain by the noise level. We have a tremendous noise level inside the nervous system, mainly because it's embedded in a biological body, which is doing a lot of weird stuff. Again, I don't have the time to get into all of this, but they're very fun, obviously. Well, let's ignore that one. So the first step is, of course, well, how can we scan the brain? And one of the fun parts we had when we had this workshop, we actually arrived at a consensus. If it turns out that it's nerve cells that are the kind of central part, we need to figure out what's connected to what and what's in them. Then we can do it with a resolution about 50 times 50 nanometers times 5 times 5. Because that way you get the smallest branches of axons. To the right here is just an optical slices through the visible man. This is what we kind of, optical resolution is far too crude to actually see everything. So the trick here is we need to actually scan a surprisingly large volume. That kind of picture that's going on to the right, that of course just represents a very low resolution picture of a very large volume. With an electron microscope you can get a ridiculously high resolution picture, but the volumes and the areas they can scan are ridiculously small. Another interesting problem is that this is very likely to be destructive. When calling it scanning, I'm kind of euphemistically labeling something that's most likely going to involve taking a brain, freezing it, pouring onto a lot of plastic, and then slicing and dicing it. And in the end, if we're lucky, we're ending up with a lot of information in a data center and a pile of dust that we kind of get rid of somewhere. But that's not going to get any customers. The reason I think it has to be destructive is that non-destructive methods, they need to scan the brain at a very high resolution over, if they're external, at least over a span of several centimeters. And unfortunately, a living brain is flopping around because the blood is moving through it, and that is very problematic. Magnetic resonance imaging is also very good. Unfortunately, even if the brain somehow magically stops flopping around because of the blood, it turns out that water molecules diffuse so much that they actually create an upper limit of the resolution you can get with MRI. One solution is to freeze the brain. All water molecules are staying in the right places, and then you can get a marvelous signal to noise ratio just by scanning it for a really long time. But again, this might not be really compatible with living afterwards. So this, of course, immediately brings up interesting question about getting volunteers and the research ethics of this and so on. But I think that can be solved. There are always people that actually think this is it would be a good idea to be a guinea pig, and as a researcher, I think that's great that they exist. Yep. So it turns out. Yep. So in general, it's actually, I was a bit shocked at our little workshop that there are so many modalities that actually managed to get with a resolution higher than the ones that we felt were needed. So, for example, a lot of the kind of traditional ones, they probably don't cut it. But electron microscopes can regularly do a resolution much better. And there are various weird methods like atomic beam microscopy, which isn't implemented. It's a kind of ultimate vaporware because we're using helium atoms to image molecules. But it really looks promising if it could work. And you can actually get down to really low levels here. The big problem, which has kind of worried me, is, of course, getting the chemistry and function out of this. But people are getting better at that. Let me first show you just an optical thing, which can be kind of demonstration that what is a training thing. Essentially, this is the training wheels version of the scanning. So this is an optical thing where you use a diamond microtome to slice off layers of tissue, and then you image them essentially with a 1D scanner as they pass by. So in the lower knife edge scanning microscopy. So this was developed by Professor Bruce McCormick down at Texas A&M. He sadly passed away shortly after our workshop, but he was very much an inspiration for a lot of this. And one of his students, Todd Huffman, has since then kind of taken it up and is starting a company doing kind of commercializing this kind of scanning. So although this might not yet be good enough for brain scanning because it's using optical resolution, which is too crude, it's still interesting because you can get very good tissue pictures. So for example, this is a mouse spinal cord they stained with India ink. So this is slice after slice. And this is also showing the interesting challenge, which I'm kind of hoping to throw at you. You can see a lot of interesting artifacts, and there's a lot of data here we want to process. And we want to get through that to produce something like this. So this is a 3D reconstruction we did. And of course, we want to do something even more than just a nice, pretty 3D picture. We actually want to get functional stuff out of it. So one of the later stuff we've done, some of you might be neuroscientists and recognize this as Purkinje cells, one of the more charismatic cells of the brain. They're completely lovely little things. And so this is the result of imaging using this microscope. But this is part of a larger data set. A much larger data set. It's actually a ridiculously large data set because it encompasses an entire mouse brain. So that's about 2 terabytes of imaging data, and it's pretty hard to display. So Todd here, he's kind of slowly zooming out and expanding the realm here. So you can see how these Purkinje cells form kind of coral reef. They're all aligned with each other, and you can't see it in this picture, but there are a lot of fibers from the granule cells going through them, making a synapse on each and every one of them, and then doing whatever they are doing down there in the cerebellum. I've heard 5 eminent researchers give their nice talks about how the cerebellum works, and there are 5 different theories, and they all sound plausible to me. We really ought to be running a simulation of this to actually find it out. So this is about how big he could show it, but he got a data set that's, well, a mile bigger than this, literally. And he's planning to kind of give that away to researchers to get them interested in using this. So this is also showing an interesting problem. Handling this amount of data is going to be a bit of a challenge. Electron scanning resolution data of entire brain is going to require an absurd amount of storage capacity, and it's not exactly clear how we could do that. So that's another fun challenge for you to think about. Even though you might not care about brain emulation itself, you might still think that there are a few good research challenges here. Over at Stanford, they have solved one of my worries about the chemistry, and that's by slicing up tissue into a little array on a microscope slide. And then you can color this using the various immunostains for whatever chemical you're interested in finding out where it is. And then you image that with a normal microscope. And now you can figure out where that chemical is. You wash off that stain, add another stain for some other chemical, image that, and then you repeat this a number of times. Now you have a map of where certain chemicals are among the neurons. And finally, you scan it with an electron microscope, which is going to do pretty nasty stuff to the tissue. But the result is that you actually get a nice topology map, as well as a slightly cruder map of where chemicals are. And the chemicals, of course, can indicate neuron type and a lot more functions. So I'm very, very hopeful about this. And of course, you're getting this in 3D, actually, because you actually have an array here of tissue slices. It's just that it needs to be scaled up a little bit, a lot of it. Then, of course, we need to do scan interpretation. We need to take these slices and stack them and actually make maps. In the past, we have been doing that using grad students. That's not cost effective. And the grad students get very bored after a while. I assume one could do something like an online game with it, but I think it would be too boring too. So this is, I think, an interesting challenge for computer vision to actually be able to take this data and actually create good 3D models of the tissue. And then, of course, you can, this is again with knife edge scanning. So they're just following blood vessels because they can't really do their full resolution. So there already exists some algorithms, but it's pretty clear that now as these data sets are started to arrive, this is going to be a much more interesting area to throw grad students at actually writing those algorithms. So I think we're going to see a lot of interesting work here. And it's also an interesting challenge managing these very, very large scale data sets with all the weird artifacts and noise. So, well, here is a kind of what we might want to actually get. We would get the little synapses sitting on the dendrites, even a hint of what their function would be. Once we got that, of course, we have a nice models, and we can actually start running traditional computational neuroscience on it. So in typical computational neuroscience, you divide a neuron into a lot of little compartments. So you can see that at a close-up, there are a lot of little cylinders. And you just simulate the electrical properties of these ones, the ion channels pumping ions in and out, as well as whatever else is going on in there. So each of these cylinders would correspond to a compartment, and you would describe it usually as a set of coupled differential equations with a diffusion term to neighboring compartments. And, well, I don't know if how interested you are in all the details, because this is kind of my main area, so I could go on absolutely forever. But the fun stuff is, of course, we have a lot of good experience here with running small simulations. We know quite well how to do that. And for certain areas of the brain, we actually have simulations which are kind of one-to-one. So, for example, the lamprey spinal cord, which is kind of a neural network of the house at my old research group in Stockholm, we actually had simulation with the same number of neurons as there are in actual lamprey spinal cords. We just didn't have a complete scan of it. Henry Markram down at ETH in Lausanne, he's doing this nice work on a cortical column. Again, he got the same number of neurons as in the real column. It's just that he haven't scanned an individual column to get his neurons. He just simulated a bunch of realistic neurons, how we would connect. And that's my kind of complaint with this model, of course. It produces beautiful activity that looks kind of plausible, but we don't know whether it actually is plausible because we can't measure what's actually going on in a real column. So we need to have these kind of scans to do that. It also, these kind of compartment models, if we use them, we actually know a bit about the time resolution and space resolution, which is useful for constraining stuff. Well, let's ignore that. So here is my old office mate, Mikael Jurfeld's simulation. So he was running this on the Blue Gene a few years back. And the interesting thing to see is he ran about 1 second and it took 5,942 seconds. And that's mainly set because you make your simulation large enough so you can get a nice coffee break while going off it. So as computers get better, the coffee break stays constant, but the simulations get bigger. But people usually don't care about the slowdown. But this allows us to make estimates about how much computation it takes per neuron. This was a fairly realistic simulation. There are other models of neurons which are more abstract, and there is a bit of debate which one should be using. From a kind of uploading perspective, we're interested, of course, in finding the most biological realistic one that does the work we want to. So, for example, Isikovich is kind of boasted with his neural model, which takes many fewer flops and it still produces pretty biological realistic modeling. Henry Markram got in a bit of spat with another researcher over this because the other researcher was running a much bigger simulation using these simplified models. And both are kind of right. Both are interesting models. So Isikovich neurons can do a lot of very useful stuff, but we're also abstracting away from biology. And we don't know how far away from biology we can do and retain whatever is important. Any question? Yeah.
Real quick, so you said the metabolic network of a cell uses maybe 5,000 proteins and we can't simulate it, but we can simulate a neuron. So why can we simulate neurons and not like regular cells?
Well, mostly it's about getting the data. And I think we're slowly working our way towards getting all the data for a cell. It's just that it takes a lot. You need to figure out what the enzymes are and their various rate constants. And that's kind of the nitty-gritty data gathering stuff. We're getting better at that, but right now people have kind of started on the important part of the metabolic network. And here is the kind of typical research problem, but you will get nice publications if you determine some constants and simulate the kind of famous and important parts, but we actually want the complete network. So for that we need the kind of automation that you also get the uninteresting parts of the metabolic network. But the kind of chemical simulation is surprisingly easy. If you want to run it, you can average things out and just assume that concentrations are enough. In that case, you get a very nice Michaelis-Menten dynamics. For some chemicals, you might actually want to think that they're discrete molecules, whereas in some cases, very few molecules or very few enzymes per cell. But that's a kind of special case, and it's relatively easy to notice.
How many moving parts does a neuron have?
That depends on how much you squint at it. So the traditional compartment model would take, for example, that Purkinje cell with about 1,000 compartments. Each compartment would have the voltage across its membrane, the state of about between 3 and 6 ion channels, and then you would get perhaps 1 or 2 internal state variables. So on the order of 10 to 20 state variables per compartment. And then, of course, we might think, well, do we need to store them as bytes, or can we simplify things further, or do we actually need really nice precision? I would be surprised if we actually needed a very high resolution. Yeah, but that's something we're going to find out. Okay. And, well, body and world model, pretty obvious that if we run a simulation, we better also give it some kind of input. So we need a body model, which doesn't really have to be quite as good as the brain model, probably. And since we're most likely to start with a mouse, it's something that's a workable mouse model. And again, you can probably do something much simpler than 100 teraflops in a completely convincing virtual reality for a mouse. Yep.
So that was my question, and this is exactly the slide where you actually address this topic. How do you argument that you're doing just a brain simulation without all of the nerves? Because, rest of the
Yeah.
nervous system in the body has some function as well, right?
Oh, yeah. For example, just grasping something actually involves not just my brain sending neural signals or a few reflexes in my spinal cord, but actually the mechanics of the tendons of my fingers actually do play a non-trivial role in making grasping work. So if I were to have a simulated arm, we would need to have at least a physics simulation here. Now the question is, how much detail do we need in this physics simulation? Obviously getting the tension in the tendons right and some of the friction stuff. And probably in order for it to feel right, we would need to do a bit of tactile vibrations. But we probably don't need to simulate the cells going on in the tendons. We can just view the tendon as a kind of abstract spring. Similarly, my bones, I don't really think a simulation of me would actually require anything other than regard them as rigid objects with a certain mass. The question about the peripheral nervous system is interesting because that's going to affect the feel. I think from a research standpoint, you might want actually to run for simulations and animal experiments, of course, where you don't really know whether it feels right. But once you get paying customers, you better get the body right. And this is, again, one of the interesting research ethics problems I haven't time to actually get into right now. But that's probably going to be a real problem. One of my nightmares is, I get cryonically frozen, then I wake up in Second Life. And I'm immortal. I can never leave Second Life. And it's never going to get better resolution. So we might want to find ways around that. Yep. Going on. Well, now, of course, as any futurist, I have to wave around Moore's law a little bit. One should always take that with a ridiculously large grain of salt, maybe an asteroid of salt. But I was kind of looking around at decade times, and we can at least plug it in to get a kind of, if we assume that Moore's law is not going to do something Kurzweilian singular very rapidly, instead just going to keep on like this or get slower, then we can actually do a rough prediction, of course, what's the earliest date we get enough computing power to do certain levels of ridiculously large simulations. It's an interesting parenthesis that if we don't need to send too much complex information, actually bandwidth is not the big problem, which is slightly surprising, I think, to a lot of people. But especially if you can just send synaptic spikes, it's actually surprisingly efficient. And another interesting thing is, of course, the differences in latency and so on seem to favor certain architectures. And then, of course, if we compare the kind of traditional Moore's law with looking at the top 500 supercomputers, you find that they advance much faster, mainly, of course, because they're actually cheating by adding a lot more parallelism. So I think commodity computers are most likely going to start catching up that rate of improvement as they get really multi-core. And then, of course, the problem is predicting that really long term. I've been having great fun recently fitting sigmoids to it, and I really understand now that we can't use that to predict very much. The kind of and the reason. So if there is anything I can kind of give you that is true knowledge in this talk, it's going to be this. If you have some kind of thing that is actually growing as a sigmoid, that would be ground truth, and you got partial data for it, and then you try to predict where the sigmoid is going to end up. You're not going to succeed very well with that unless you're just having great luck until rather far into the process when it's self-evident anyway. And the reason is at the early stages, you're essentially just seeing a straight line or maybe something vaguely exponential. So the uncertainty and eventual level is going to be ridiculously large. And in this case, of course, we're looking in logarithmic space, so this you need to exponentiate the uncertainty. So I get 11 orders of uncertainty in my predictions here. We can be pretty certain, I think, with a 95% confidence interval, that we're going to get at least a factor of 100,000 more computations per dollar. That's the pessimistic scenario. The optimistic one is one where I think we're actually running into issues of quantum physics. It seems that, no, you can't squeeze that much information to the solar system. But I think that's about 10 to the 27 computations per dollar or something like that. I simply don't believe that. But the kind of middle range, we're going to get ridiculously much more computation. And then just assuming a simple Moore's law and counting how much state variables and stuff would we need on the different levels. I don't know whether any of you can actually read this particular font, but I can always put this online. We actually got a white paper with it. So you can actually plug it in and see, well, the earliest year you could get that. So for example, if we want to run an electrophysiological simulation of a brain, where we have this compartment with about 10 state variables, 1 byte for each, in that case, you end up with a memory requirement of about 10,000 terabytes, which you could do for $1 million about 2019, assuming everything keeps up. Obviously, if you got more money, you can do it earlier. And if you need to, for example, store a full 3D map using electron microscopy, then you need about 1 billion terabytes, and that's going to be affordable for a millionaire about 2043, assuming the cost of memory and so on keeps on. And of course, if it's quantum, then we just need to replace with qubits, and we have no clue. But it's interesting, we can already put a bound here. Depending on where reality actually lies, we're going to be able to do it at certain times. Or rather, that's the earliest time we can do it affordably, because later on it's going to be cheaper. Then, of course, we can look at the computational demands, and we can similarly plug things in here. And it turns out that if the brain were just behaving like a simple perceptron network, we could actually probably run it, or actually can run it, they could run it on a supercomputer today. It's just that I don't think anybody believes that. You know, if you want to run this with electrophysiology, well, between 2033 and 2068, around there, it starts to be kind of computationally feasible for $1 million of computing equipment. And then, of course, it gets more and more extreme. And once you need to actually do a nuclear simulations, it produces an estimate of 2,200. And at that point, we can just say, OK, somewhere indefinitely far or probably never. So if it's feasible, we can still plug this in and make kind of a graph and start looking at it. So this is probably also very hard to see. But depending on the green line, the horizontal green lines represent the different estimates of what resolution we need to run it. And the blue and red line represents the first time you get enough computing power for that. So you can see the intercepts that represent kind of earliest stage. The problem is, of course, all this depends on us neuroscientists doing our job and actually understanding enough of whatever level we want to simulate to actually do it. And we have no guarantees about that. Science never really produces good timetables when it solves problems. Sometimes it does it in the wrong order and the wrong time. The interesting part is scanning seems to be almost there already. If we know what level we need to scan, we can probably do it. It's just that we need to scale up the scanning equipment. We're probably going to need an enormous factory of electron microscopes or something like that. Kenneth Hayworth has some really interesting work on this for doing it. But that can be done. So one of the interesting things is, of course, well, if you got a lot more money, you can do it earlier. So if you're looking at computing power, you can actually accelerate it between 1 or 2 decades if there is regarded as, oh, this is really important. And the most interesting thing is, well, smaller brains. Obviously, we can probably run an insect brain today, and that would be a very worthwhile research project. And the mouse brain, I think, is important because at that point, people are going to start realizing, oh, in a few years, we're going to be doing humans. Oh, social impact, first mover advantage, strategic importance. And at that point, money is going to start flooding in. So from an ethical standpoint, if it turns out that brain emulation is a really bad thing, we ought to figure it out before that. And then start somehow making the field embarrassing enough so nobody works in it. If we think that this is great, this is going to solve the world problems, on the other hand, we really should work very hard towards that mouse model. And it still seems to be a surprisingly short distance. The really hard scientific problems about scanning a brain, making a simulation, creating this pipeline, that's solved by the point you get to the mouse. Once you have a mouse, then it's just a matter of scaling up, which is non-trivial, but it's still relatively clear engineering. And that's slightly scary because at that point, 20 years might be optimistic. Well, and the order actually does matter. Because if we think that the kind of surprise, whoops, suddenly we got an enormous change in world economy and the human condition, that might be destabilizing. Then we should really make sure that we get the scanning and neuroscience done, and then we can just trust Moore's law. Because it's unlikely that you can make Moore's law work faster. And that's such a big, powerful thing that is run by a lot of other interests in our economy, but that's not going to be affected. The scary possibility is that, well, neuroscience is not doing anything good, and we're having nice scans. We're all over data centers, and researchers are poking around in them, and the computers get better and better. And then one grad student says, wow, we forgot about simulating this. And, well, now we can go from mouse to human in, well, whatever time it takes to dig up a good human model. That would be very destabilizing and scary. So we need to keep an eye on this. But I think this is going to be a very interesting area. And, well, since Google wants to organize all the data around, and I think this neural data might be very interesting to organize. I haven't time to get into the energy requirements and footprint, but I think it's actually a software humanity would possibly have a very small environmental footprint. So I would like to say thanks to the people who helped me develop this, and thank you for listening. And we got a white paper over there if you're interested in more detail. Questions? Yeah, well, briefly, yeah.
So where are we with regard to simulating C. elegans or some simple organism, body model, whole thing?
Body models of simple organisms, I think we're doing decently, at least if you want something snake-like or lamprey-like, actually, we have sufficiently good models to do it. The C. elegans neural network, the problem, as I said, is we don't know the science of the synapses. I don't think anybody's actually going to do that research because it's just too tricky. I know some people like Peter Passaro down in Brighton in Britain who really wants to get started if we can get funding on a project on scanning the British pond snail, which is very nice because it's got big neurons you can figure out and they're nicely compartmentalized into different ganglia. And I think computationally we could definitely do that. We just need to figure out exactly which neurons are connected to what and what the properties are.
How much money does it cost?
I would guess for a large-scale project, I would guess about £1 million or something like that. It depends a bit. It also depends a bit on what research system you want to start with. So, for example, another area that we could perhaps start scanning would be the retina, which would be very, very useful, but not very glamorous. Everybody wants to have cortical columns because they're the spacey and cool thing. And if we figure out what the cortex was doing, maybe we're going to solve some of the tricky problems for AI and machine learning. But the retina, for example, doing a retina scanning project would also probably not be too costly. I would guess about $1 million or something like that. I'm just guessing about the costs here. But I think in general, we're surprisingly close if there was just somebody getting the funding and deciding to do it. So, yeah. You first, and you.
So I'm wondering, have you talked about people who do that?
Summarizing this question for the recording is going to be interesting. But yeah, can we find the part? Let's see. What subparts of the brain are? What you describe is essentially the issue of how to do an economic simulation of a very complex system. And ideally, you want to figure out what's going on, and then you can simplify the simulation. You don't need to run the full electrodynamics of a transistor. You can just replace it with a logical element. But you probably need to run the full dynamics first in order to figure that out, especially if you're trying to reverse engineering, let's say, a Pentium 4 processor this way. So this goes back to the issue of scale separation. And I think we have decent ideas that there must be some scale separation in biology, because if you don't have that, if there is a lack of modularity, it becomes very hard to actually evolve anything. And neurons are pretty nicely compartmentalized. They're electrically quite insulated from the surroundings. We certainly have an interaction with the surroundings, but it's relatively local. And similarly within cells, you again get a lot of compartmentalization. I think that is what I would be kind of using in order to simplify things. But what I'm interested from a kind of general scientific perspective is actually, could we develop good tools for rapidly figuring this out in a new system? Well, how low level do we need to do the simulation on? And sometimes you can figure that out by looking at time constants, for example. And this scale compartmentalization, or the physical compartmentalization, also does this. The tricky thing about a chip, for example, is transistors have very long-range connections. They're not just local. The brain is actually quite nice in this regard, because a lot of all the interactions tend to be very local. But yeah, I'm happy to discuss this later. I think it's a bit tricky to quickly answer. So it was a question over here. Yeah.
Yeah, why did you choose $1 million as the amount of resources you would spend on this?
That was because of a paper by Robin Hanson. He points out that if it costs $1 billion to make a brain scan and run it, well, now great, Bill Gates gets immortality or at least a copy. But once you're down to about $1 million, now it becomes cost effective to actually copy professionals. And that is economic plutonium.
It seems like the point of making a timeline is to anticipate the thing happening so you can make ethical decisions. It's very important to have a good economic model. Do you have that?
Partially. Robin Hanson's model is the only one that currently exists. Among my friends, we're having kind of informal models that are slowly working their way towards actually becoming real papers, mainly because we're so scared about Robin's very cheerfully cynical model that seems to suggest a population running off towards infinity and wages going down towards zero, which seems to be rather dystopian. But yeah, there is an interesting arbitrariness about the money, and obviously the first project is going to be some kind of, well, if not Manhattan project, at least a well-funded research foundation, maybe the Allen Brain Map or something like that. Yeah.
So the idea of doing brute force scanning seems kind of a lot of work. It seems like if you have the genome, if you have a suitable model and simulation of physiology and neurogenesis, you should be able to come up with the brain model from that. Do you see a point where that would actually be possible and you could bypass the whole scanning?
Well, neurogenesis is interesting, but we actually don't have much experience in simulating development of organisms yet. And that is a real problem. If we had a good way of growing brains in the software, I think that would be very good. But right now, that's a much less developed area than this kind of neural network models. It might turn out, of course, that we're going to get a few surprising breakthroughs in the next decades. And in that case, that might be a better way of doing it. That would be a way of getting artificial intelligence kind of by cheating. Oh, it's actually a kind of a human child growing up inside a computer. Again, a lot of interesting ethical questions that are going to keep me and my colleagues nicely salaried. But from a kind of developing AI perspective, yeah, it might work. I'm suspicious that it's going to be, I think it's going to be problematic because the biology actually in development is really messy. If we're a bit concerned about the messy biology of an adult brain, the development is going to involve structural remodeling. Again, a lot of local interactions, but a lot of this is going on in low concentration, morphogens diffusing around, and that might be pretty messy. But I'm hoping to stand corrected. Yeah.
You talked about dynamic states of the system. Do you consider sleep, consciousness and sleep as a particularly interesting sort of, state change, or are you not really heading out with it?
Well, I think that is a kind of high-level difference. What's going on kind of low-level to drive it, I think we have a decent understanding of in terms of noradrenergic activating system and so on, kind of regulating which kind of attractor states the brain tend to move into. So sleep and consciousness, well, I think a brain simulation of sleep, that's very doable. Actually, I've seen some papers about that. Whether we would get consciousness is a completely different matter. And given that I get horrible headaches from philosophy of mind, I'm kind of trying to avoid that. Yeah.
Ask if any of the VC sites have questions.
Yeah. Any questions from the other sides? No. Local questions? People have better things to do. Thanks.